So, epigenetics. So, this is a blooming field, and I mean, the way epigenetics is conceived has evolved since its first definition, as you know, by Conrad Waddington in the late 30s, the beginning of the 40s. And so my aim today is a descriptive aim, in fact I want to show that epigenetics is a multifaceted research area, and that its diversity in biology, in different biological disciplines, is due to its historical evolution. And so I try to, in a certain way, to address a sort of typology of conceptions of epigenetics, trying to find out its origins, two origins maybe, and trying to identify some traditions of the way epigenetics is conceived. And what I want to show also is that epigenetics first has risen as an answer to the problem of development, and during its evolution it has shifted towards an answer to also evolutionary issues. And finally, always referring back to Waddington's idea, epigenetics seems to me is a sort of bridge between development on the one side and inheritance, or development and evolution. And it's a sort of, I would say that it's not really a discipline, but a sort of perspective, integrating different disciplines, merging together different disciplines and different issues, from development to inheritance and evolution. So here is the plan of my talk. I will first talk about epigenetics and the problem of development, then, and so I will introduce some conceptions of epigenetics. Then the second part is about epigenetics and the problem of the origin of the developmental origin of phenotypic variation and evolution, and two other conceptions, and then I will try to conclude with some general considerations about all these conceptions. So first, epigenetics and the problem of development. So Waddington, I go there because I cannot see what I wrote on the slides. So Waddington, the question he was interested in is the question of the development, of how another organism arises from the individuals of the previous generations. And he wanted to answer to this question by putting together, by merging the results of experimental embryology of his time, conceived as developmental mechanics, and the results of genetics, the science of inheritance. He wanted to merge, because in fact experimental embryology investigated development, doing experiments on its mechanisms, and genetics looked on the phenotype in order to make inferences about the units of inheritance and the mechanism of inheritance. So genetics explains development in terms of changes in the units of inheritance, gene changes. And so the problem for him was the way genes have an effect on the developmental mechanisms of the embryo. So it's in 1939 book, An Introduction to Modern Genetics, that he started to, sorry for the noise, started to conceive, qualify development as an epigenetic process. And he wanted really to understand what lies between the phenotype and the genotype. How, in particular, the genotype can bring about phenotypic effects. And so the answer, in these terms, is that the epigenotype lies between the two, or the epigenetic constitution of the organism. And he defines the epigenotype as the whole complex of processes which dynamically organizes and constructs tissues, organs, and then the entire organism by interacting with the genotype and reacting to the external environment. So interactions between genes and between genes and the environment. And as you know, 1 year later he introduced he's sort of, it's not really a model, it's a metaphor in order to talk about what happens between the genotype and the phenotype, what is such an epigenotype, and later, many years later, he introduced more precise pictures of what he calls the epigenetic landscape, and here you can see the genes that interacting each other and with the environment in a certain way form the epigenetic landscape. And it's in his famous paper in 42 that he defines what he means by epigenetics. He defines it as the study of the causal mechanisms at work in development by which the genes of the genotype bring about phenotypic effects. So in fact, epigenetic is the study of the epigenotype. And Waddington really consisted at the accomplishment of his project to merge research in experimental embryology and research in genetics. So, if I take it here, it works. I mean, you heard me? So, in fact, is to merge the explanation of development both in terms of the action of organizers, so in terms of what experimental embryology talked about and explained development, and in terms of genes action. And it's also in this paper that he explicitly say that he chooses the term epigenetics in order to focus on the classical theory of epigenesis. Epigenesis, so it's the idea that the organism is not preformed in the zygote and then just unfold during the ontogenesis. On the contrary, the organism is progressively constructed during development by the interaction of the constituents of the egg. There's no preformed organism in the zygote. And so I want just to precise that he did not use the prefix epi to mean acting above the level of the gene. He wanted to mean to refer to the theory, the ancient theory of epigenesis, which dates back to Aristotle. So let's come back to the problem of development. So how is another organism progressively constructed from a fertilized egg? And so this problem, as you know, include the more specific problem of cellular differentiation via the regulation of the gene, the action of genes, and later, starting from the 60s, the activation of genes. And so this was, the problem of development was the main worry of Waddington. So more precisely, the problem is that how do cells of the same genotype can express differently their genes and so acquire distinct phenotypic features, giving rise to different types of cells and then tissues and then organs. And so just quickly, some answers, some models in order to answer to the problem of development, because Waddington's was not really a model, as I said, it was more just a sort of metaphor in order to answer to the problem of development. So as you know, in the 50s, Barbara McClintock suggested the hypothesis of coordinated transposition of elements controlling gene expression, so transposing elements controlling the expression of genes. But this hypothesis was later discarded, in particular by experiments showing that showing the developmental capacity of nucleus coming from differentiated cells. Then you have in the 60s the overall model of the regulation of gene expression in prokaryotes by François Jacob and Jacques Monod. And so it's, the model is based on the idea that a set of structural genes are regulated by a set of adjacent regulatory genes. And this model was had a great success, but at the same time it was criticized by researchers because it was not really able to explain global changes in gene expression in a cell. It just accounted for progressive modifications of gene activity via a cascade of regulatory genes activation. And so came in the 8 years later, the Britton-Davidson model, also called gene battery model, which was proposed in such a way as a criticism to the operon model in order to fulfill some limits of the operon model. This model is at its core, that's the idea of gene networks, which are involved in the simultaneous activation of a huge number of non-contiguous genes. And so you can see that here we have, if you know this model, I am sure that you know it, you can account for global changes in expression in the cell, simultaneous activations of a large number of genes. So let's continue and come back to epigenetics, come back some years in the late 50s. So another biologist, in particular Celiatologist, tried to use the term epigenetic in order to answer in a certain way to the problem of development. And so David Naney distinguished between two control systems, genetic control systems and epigenetic control systems, which are both involved in determining cellular characteristics. So genetic control systems, their hypothesis was based on the recent research advances at Naney's time about the physicochemical structure of DNA and about the fact that DNA can be replicated in a semi-conservative way, and so there's a sort of memory through cell division of the information contained in the DNA sequence. So genetic control systems, their aim there is to conserve the library, in terms of the library of specificity, no matter whether these specificity are expressed or not along cellular lineages. But according to Naney, you have encoded information in the DNA, but you need something to decode this information. And so here is the hypothesis of epigenetic control systems, which are there in order to regulate the expression of genetic potentialities. They are auxiliary systems to the core genetic specificities. And so this is a theoretical answer to the problem of development and cellular differentiation. And the central feature of Naney's hypothesis is that both epigenetic and genetic control systems are mitotically stable. There's a cellular memory of these systems. And this is a big difference with Waddington's conception of epigenetics, who didn't explicitly talk about some cellular memory or some cellular stability. So it seems to me that Waddington and Naney, they both aimed to answer to the problem of development, trying to integrate genetics, classical in the case of Waddington, molecular in the case of Naney, with developmental biology. But the difference between the two is that there was the rise of chemical genetics, molecular genetics, in the 50s, and so they tried to answer to the same problem at different levels. So then in the 60s and the 70s, I'm going very quickly, some theoretical and experimental advances in molecular biology are very important in the history of epigenetics, it seems to me. Two of them are particularly important. First, the discovery that chromatin states influence gene activity, in particular researches made by Mary Lyon in the 60s and 70s, and then the study of the DNA organization in nucleosomes. And thanks to these advances in molecular biology, the problem of development was open to receive a more specific molecular answer. And so we come here to Rigg and Holliday, who in the 70s, in 1975, they independently suggested the hypothesis of the regulation of gene expression by DNA methylation. And here we can find again this idea of mitotic stability. In Holliday terms, it's so the fact that DNA methylation is somatically heritable. And so at this time, in their respective papers, there is no mention of the term epigenetics. This mention came later. in the paper by Holiday. He used the adjective epigenetic in the paper in 1979. And so here you can find really a molecular, even intracellular answer to the problem of cellular differentiation and more generally of development. And so this is another conception of epigenetics. In the 90s, you start really to find many reformulations of this conception of epigenetics. Here you have one by Holiday in '94, and then one of the most quoted definition by Russo and others. So I read it. The study of mitotically and or meiotically heritable changes in gene function that cannot be explained by changes in DNA sequence. So I just to note that at the beginning, Ray and Holiday talked only about mitotic stability, but then later they, Holiday in particular, he added the idea also of meiotic stability, and then you can find this idea also in many definitions of this kind of epigenetics at the molecular level. And so if we have to identify some defining features of this conception of epigenetics, there are three. Cellular stability or memory, as I said, both mitotic and meiotic. The impact on epigenetic modification on gene expression or gene function, because talking with Christophe, he made me notice that sometimes the definition refers to gene expression, sometimes to gene function. It's not the same thing, and so it could be really problematic if you to refer to one or to another, and to talk about gene function referring to gene expression and the other way around. So. And the other feature is the fact that epigenetic modifications don't involve modifications of the DNA sequence. So I would say that Rick's and Holiday's conception of epigenetics is part of Nanney's tradition of how epigenetic is conceived, because in a certain way you can find these three defining features also in Nanney's conception. Even though it was more difficult for him to test these ideas. There's the idea of cellular memory in Nanney's definition. There's the idea of an impact on gene expression, because genetic control systems decode in a certain way the genetic specificities. And there's also in a certain way the idea that the changes in DNA sequence are not involved, even though Nanney thought that epigenetic factors were mainly in the cytoplasm and not in the nucleus. So this is one difference. And molecular epigenetics today, in the same tradition, it seems to me, we can find today a sort of a relaxed conception of epigenetics with respect to Rick's and Holiday's, in which the stability or the cellular memory is no more a condition sine qua non of epigenetic changes. So we can find looser and more inclusive definitions of what epigenetics is today in a molecular context, in which any chromatin modification affecting gene expression is considered as epigenetic, regardless of its stability or heritability. Because we try to show that there's some memory, some stability, but in some cases we have not yet succeeded to show that. So just to make a short break, this is in a certain way what I have said until now. Waddington's conception of epigenetic, Nanney's conception, Rick's and Holiday's, and the current molecular conception of epigenetics, all trying to answer to the problem of development. Now we come to the part about epigenetics and the problem of the origin of phenotypic variation in development and the problem of evolution. And it seems to me now we have a sort of shift from developmental issues to more evolutionary issues. First of all, what I call the Evo-Devo epigenetics. So as you know, Evo-Devo, it has emerged evolutionary developmental biology in the 80s, and so it's a research field interested in the relationship between development and evolution. In particular, despite many different sub-traditions in evolutive biology, the main focus, the common focus, is the genotype-phenotype map, and so the problem of how phenotypic variation, phenotypic novelties emerge during development, and of the impact of such a phenomenon on evolution. There are various conceptions of epigenetics in the evolutive literature, and they are all explicitly inspired by Waddington. They refer to Waddington all the time. So the question is, do they belong to Waddington's tradition, which would be the second origin of epigenetics, with none the other origin. David Hyde wrote a paper in 2004, a very nice paper, saying that the two origins of epigenetics are Waddington's conception and Nanning's conception. I agree with it. So first, very quickly, narrower conceptions in the evolutive tradition. They are focused on intracellular mechanisms, altering gene expression, and so the phenotype. And here you have an example of this kind of definitions, not so far from the molecular definitions of what epigenetics is. So the differences are that there's no explicit mention of the stability, of the heritability of epigenetic mechanisms, but I think it's implicit also because evolutive people are interested in evolution, and so you need some stability in order. Right? You need variation, but you also need some stability, some heritability in order to have evolution. Gene expression is explicitly mentioned, and you can see the focus is phenotypic variation, the origin of phenotypic variation. And so we can see already a sort of the beginning of a shift from strictly developmental issues to the problem of how the phenotype changes and then evolves. But the more interesting, according to me, definitions in the evolutive tradition are broader conceptions of epigenetics, covering many levels of biological organization, and so referring to a huge number of, a huge diversity of mechanisms. So here you have a quotation from the book edited by Hallgrimsson and Paul, called Epigenetics. And so I have a longer definition later if you want to see it. I have no time to show it. But you can see here that there is no mention of stability, heritability, or epigenetic mechanisms, like in the case of narrower definitions. There's no explicit mention of the process of gene expression. And that also there's the same shift from developmental issues to more evolutionary issues. In fact, if you read the introduction and the conclusion of the book and the entire book, you can see that this conception of epigenetics, why I say that it covers many levels of biological organization, because they define, they qualify as epigenetic every interaction that has an impact in a certain way on the developmental process and the origin of phenotypic variation. So they start from the molecular level, referring to modifications of chromatin marks, going through, for instance, the cell level, for instance, interactions between two populations of cells, giving rise to a third population of cells is considered as an epigenetic mechanism. Also, they talk about epigenetic at the level of tissues, at the level of organs, until the level of populations and species. For instance, the interaction between populations of prey and of predators are considered as epigenetic interactions, epigenetic mechanisms. For instance, when a predator, in a certain way, puts in the environment some chemical element, and this has an impact on the prey, but they consider this as a case of epigenetic mechanisms. Now, we saw that at the beginning, one important element, a sort of necessary condition for development to take place is mitotic stability or cellular memory, in order for cells to keep along their lineages some differentiated state. Then we saw that in the 90s, also in the definitions of epigenetics, we can find also meiotic stability, and not only mitotic stability. And so this means the possibility of between generation transmission. So via mitosis in the case of organism with asexual reproduction, and via meiosis in the case of organisms like us. And so these can have important consequences. It's one of the questions you asked to yourself, I imagine, on the way we conceive evolution, and on the way we describe and account for it by the evolutionary theory. So consequences of heritable epigenetic modifications for the evolutionary theory. And so many people are talked about an extended or an expanded evolutionary theory which would integrate advances about epigenetics, in particular transgenerational epigenetic inheritance, but also integrating other forms of non-genetic inheritance. And this leads me to the last conception of epigenetics. I call extended synthesis epigenetics because of this objective of finding out, of these people finding out an extended or expanded synthesis of evolution. And Jablonka and her collaborators, Jablonka and Jablonka, is a paradigmatic case of this kind of conception, which is, it seems to me, a sort of mix between Nanni's conception and Waddington's conception of epigenetics, because in her definitions she refers to the idea, really he used this expression, epigenetic control systems, as involved in cell determination and differentiation by regulating gene activities without altering the DNA sequence. And she also refers to the idea, Waddington's idea of the epigenotype. So the set of mechanisms which dynamically construct the organisms through development. So here you have a definition by Jablonka, for instance, and you can see that she refers to persistent developmental changes. So she keeps the idea, which is very central, of cellular stability or memory, which is so very important for development. And then she focused on transgenerational epigenetic inheritance. I go quick here. She distinguishes two senses, a broad one and a narrow one. So the broad sense of transgenerational epigenetic inheritance is inheritance transmission through developmental interactions, body to body, like through mother-offspring interaction, through social learning, through communication. And so she put it aside, and she focused on the narrow sense of transgenerational epigenetic inheritance, which is through cell-to-cell interaction, in particular through the germline, through this bottleneck in sexually producing organisms. And why does she focus on this particular element of epigenetics? Because, as you know, her objective is to show that advances in epigenetic research represent a challenge to the traditional evolutionary theory of modern synthesis, and so that we have to look for an extended synthesis integrating all these advances. So the objective is no more development per se, but it's more this searching for a new synthesis. There are other people, of course, in the same kind of research tradition. So in this case, the problem of development merges with evolutionary issues, and epigenetics becomes a sort of means to challenge the traditional view of evolution, to address the question of how the traditional view of evolution should change, and to profoundly rethink it with a view to this new synthesis of evolution. So you have here the complete all the definitions, all the conceptions I have just introduced. So the two at the bottom are part of this conception of epigenetics as looking also for an answer to some evolutionary issues, with the extended synthesis epigenetic as the one who is most on the side of evolution. Of course, I want to precise that it's not, there could be other categories. Maybe you don't think you belong to one of these categories. It's just a typology I'm trying to make. And it's a continuum. It's not that you have to be in one of these categories. It's just a continuum. And so to conclude, there could be two possible scenarios about all these conceptions. There could be two traditions, but you could distinguish two traditions in different ways. The first way is to do as following the plan of my talk, saying there's one tradition of epigenetics looking for an answer to the problem of development. And so you put Waddington with Nanney, Riggs and Holliday, and the molecular current conception of epigenetics. And the other tradition would be a tradition looking for the problem of the developmental origin of phenotypic variation and the problem of evolution, and so the Evo-Devo and the extended synthesis epigenetics in the same tradition. So it will be something like this. So in blue, the developmental tradition, and in yellow, the other tradition, with the extended synthesis epigenetics in green, because it's a mix between the two traditions. But I will be more for this distinction of the two traditions, with Waddington as one origin and Nanney as the other origin. And in the case of Waddington's tradition, the heritage is the common approach, in part the approach of developmental genetics, even though the approach is broader now in the case of Evo-Devo and the people working on the extended synthesis. And the main idea, the main heritage in terms of ideas is the real importance of developmental interactions in development and also in evolution. And this idea of epigenesis, the fact that there is no preformed organism in the zygote, but development is, the organism is constructed throughout development by genes interactions and by the interaction of all the different components of the egg. And in the case of Nanney's tradition, the common approach would be molecular genetics, and the focus is on chromatin modifications and their impact on gene expression. And here, it seems to me that we can discuss about that. There's still the idea of a developmental program, a sort of preformationism. The idea that there's some information in the DNA sequence, and then depending on epigenetic factors, this information is differentially expressed in time. And so the picture would be something like that, but it's not so evident to see the first time. So what about you? I'd be interested in order to know where do you situate yourself, and whether you situate in another sort of conception of what epigenetics is. As I said, it's a continuum. And why is it a continuum? Because it's a continuum between development and heredity, development and evolution. I remember that Waddington wanted to merge experimental embryology and genetics, and in order to find out an answer to the problem of development. And we can find this duality of development and heredity, development and evolution throughout the history of epigenetics. And so if you are more interested in development, more on the side of development, you put forward the idea of cellular stability, of mitotic stability. This is what you are interested in. And if you are more on the side of evolution, you are more interested in transgenerational epigenetic inheritance. And epigenetics also, it seems to me, is a sort of bridge, as in the case of Waddington, a bridge integrating disciplines. In the case of Waddington was experimental embryology and genetics. In the case of Nanney and all the molecular tradition, It's a bridge between molecular biology and developmental biology. And in the case of the evo-devo epigenetics and the extended synthesis epigenetics, the bridge between developmental biology and evolutionary biology. So these are all the reasons why I will not say that epigenetics at this moment is really a discipline. It seems more a useful perspective in order to find out answers to different problems. It depends on the discipline you work in, and so it could be the problem of development, the problem of evolution, and also and the two aspects of development, inheritance, evolution, three aspects in this sense, because heredity has a lot to do with evolution, so it's on the other side. So to integrate these different aspects and to find out more complete answers to all these problems. So more a perspective rather than a definite disciplines with just a unique project for it. So thank you very much, and thank you to Antonin Nicodou, who is my co-author. We are working together on a few papers on this kind of topics. Thank you very much.